Welcome to the deep dive. If you're here, you want a fast, thorough shortcut to being well-informed. And today we're doing a really focused analysis on the biology of, well, systemic aging.
We have a great stack of sources here. They all get into the mechanisms of rejuvenation. Specifically, we're looking at what's circulating in your blood and how manipulating those factors could, actually turn back the clock.
It's really the holy grail of longevity research, isn't it? This idea that aging isn't just a passive decay.
It's not. It's an active process driven by some pretty malicious cellular changes. And our focus today is on one of the big ones, cellular senescence.
Which is pretty much accepted as a major hallmark of aging.
A major hallmark, exactly.
Okay, so let's unpack that. Senescence. We're talking about cells that stop dividing, but they don't die. They just sort of hang around.
Right, they become dysfunctional. But the really amazing part from these sources is that they don't just sit there quietly. They start broadcasting a toxic signal.
A toxic language, almost.
That's a great way to put it. We call it the senescence-associated secretome phenotype.
SASP.
And you can think of it as cellular pollution. It's a mix of pro-inflammatory cytokines, things like IL-6, IL-8, GDF-15.
And these things spread the toxicity.
They do, like wildfire. It hits neighboring healthy tissues, and that systemic inflammation promotes basically every age-related disease you can think of. Cancer, fibrosis, you name it.
So the goal is clear. Either silence these cells or get rid of them completely.
Which leads to two main strategies we see in the research.
First up are the senolytics.
Senolytics, that's the kill and clear strategy. You just eliminate the senescent cells.
The scorched earth policy.
Kind of, yeah. And then you have the senomorphics.
Which is a more nuanced approach.
I would say so. Instead of killing the cells, senomorphics just suppress that bad behavior. They basically flip the off switch on the toxic SASP factors.
So you render the cells harmless.
Exactly. You silence the pollution.
So today we're digging into how researchers are tackling this problem systemically, through the blood. We'll look at the subtractive approach, literally removing old, toxic stuff.
And then the additive approach, introducing concentrated young restorative factors. Okay, so the subtractive approach. The foundation for all this really goes back to those almost infamous heterochronic parabiosis experiments.
Right, where they stitch old and young mice together so they share a circulatory system.
Exactly. And for years, the entire scientific community just assumed the rejuvenation they saw in the old mouse was because of, magical youth factors from the young mouse.
But that wasn't the whole story. The sources point to a huge paradigm shift with the neutral blood exchange experiments, NBE.
This was the critical proof point.
So walk us through that. How did NBE challenge that old assumption?
Well, NBE is basically a tiny version of a human procedure called therapeutic plasma exchange, or TPE.
Which is already FDA approved for things like autoimmune disease, right?
It is. So in the mouse studies, they did this NBE. They removed about 50% of the old mouse's plasma. But, and this is the key, they replaced it with just a neutral solution, saline and albumin.
Wait, so no young factors were added at all?
Not a single one. And that was the revelation. The rejuvenation happened just from fleshing out the old components.
That's incredible.
It totally shifted our understanding. It proved that aging is largely driven by these age-elevated inhibitory systemic proteins.
Stuff that builds up over time and actively holds the body back.
It actively suppresses tissue health and repair, so we don't necessarily need to add good stuff first. We need to remove the bad stuff.
The body is being held back by its own accumulated junk. So what happened in the mice when you took that junk away?
The results were immediate and they were widespread. After just one procedure,
Wow.
you saw statistically young-like outcomes. Muscle regeneration improved dramatically. Fibrosis went down. In the brain, they saw more neurogenesis.
The creation of new neurons.
And way less neural inflammation. The liver got better too. Less fat, less fibrosis.
There's a natural resilience there that's just amazing. You mentioned a second punch mechanism. So if you remove the inhibitors, the body's own youthful proteins, which are still there but just suppressed, they can bounce back.
That's the magic of it. The body's own youthful proteome, it wasn't gone. It was just muted, like a radio station with a ton of static.
So you clear the static.
You dilute the inhibitory factors, and the body's natural signals, things like neuroprotective brain proteins, can become elevated again. The whole system gets a chance to reset and heal itself.
Okay, so let's bring this to humans. Therapeutic plasma exchange, PPE, is already a thing.
Yeah.
And a clinical study actually looked at its effect on biological age using epigenetic clocks. What did they find?
That study was a huge clinical validation for this whole subtraction idea. It showed TPE led to an average 2.5-year reversal in biological age.
2.5 years.
And what's interesting is the biggest effect, about 2.61 years, was when they reinfused immunoglobulins, the body's own antibodies, as part of the procedure.
So this proves that a clinically available method, just by diluting bad stuff, is a real strategy for reversing measurable aging markers.
It's a viable strategy, yes.
Okay, so removing the bad works. That's huge. Now let's flip it. The additive strategy. Can we get a similar, or maybe a more targeted effect
Right.
by adding concentrated good factors back into the system?
Right. And this takes us into the world of stem cell-derived secretomes and extracellular vesicles, EVs.
Sometimes called exosomes.
Exactly. These are incredibly tiny, nano-sized particles that cells secrete. Stem cells are especially good at this. One source focuses on human trophoblast stem cells, or HTSCs.
And this is very early stage, ethically sourced material.
Correct.
So these EVs are like a perfect little nano delivery vehicle. What are they carrying?
Think of them as membrane-bound bubbles full of bioactive cargo. You've got proteins, messenger RNAs, and what seems to be most important here, specific microRNAs.
They're like secondary messengers.
That's what they are. They travel to a recipient cell and directly change its gene expression.
So what happens when you apply these HTSC-derived factors to senescent cells?
You get a powerful senomorphic effect. It's a huge suppression of that toxic SASP signal.
IL-6 and IL-8 levels drop.
They drop dramatically. And the mechanism seems to be that the EVs are delivering crucial proteins that are missing or downregulated in the old cell. It seems to restore lost functions, like improving the cell's ability to repair its own damaged DNA.
That's a powerful shotgun approach. But the research gets even more targeted. It zeroes in on what they call the E5 miRNA cocktail.
Yeah, this is where it gets really fascinating. They identified 4 specific microRNAs that were enriched in those stem cell EVs.
And they had to be used as a cocktail, right?
Yeah.
Because individually, the results were mixed.
That's the complexity of it. Individually, some might reduce one aging marker but actually increase SASP inflammation. You have to find the perfect combination.
So when they put the E5 cocktail together, how well did it work?
It worked as a true senomorphic. The combined cocktail suppressed all the measured markers of senescence P16, P21, SALT-Gal, and the inflammation markers like IL-1s and IL-6.
And this worked in living animals.
It did. They packaged it into liposomes, delivered it to naturally aged mice, and saw reduced senescence and inflammation in the liver, kidney, and spleen.
So let's get to the molecular target. How does this precision strike actually work? You mentioned the P53 signaling pathway, PCAF, and HIPK2.
Right. Think of the P53 pathway as the cell's main stress manager. It makes life or death decisions. And PCF and HIPK2 are like 2 key input levers that control how aggressively that P53 pathway turns on the toxic SSP program.
So the E5 cocktail targets those levers.
Precisely. It suppresses PCA and HIPK2. By turning down those 2 inputs, you stop the P53 pathway from generating that toxic inflammatory mix. You shut down the core machinery.
That really is a precision strike. So we've gone from wholesale blood exchange to precision miRNA cocktails. Let's touch on 2 other ideas for scaling this, starting with something non-human, the Porthian E5 fraction.
This came from another research team, and it confirmed the same thing. The rejuvenating factor in young plasma isn't small molecules, it is the nanoparticles and exosomes.
Which reinforces the whole last section.
It does. So the challenge then becomes, how do you scale this up?
And that's where pigs come in.
That's where pigs come in. They tested a fraction from porcine, or pig, plasma. And the logistics here are pretty compelling. There are 1.4 billion pigs slaughtered every year. Blood is an industrial byproduct.
So you could make a globally accessible product.
Potentially, yes. Much quicker than with human-sourced materials.
And it worked across species.
It did. The porcine E5 fraction worked in rats. And maybe the most remarkable part was a topical application on a human hand. The anti-aging effect persisted for over a year with no bad side effects.
Okay, from biologics to synthetics. Let's talk about MXB22510. This is a small molecule designed to mimic a key human peptide.
Right. It mimics a naturally occurring peptide called LL37.
And LL37 is really important.
It's crucial. It's involved in systemic healing, recruiting stem cells, managing cytokines, and, critically for our conversation, inducing apoptosis or programmed death in aging cells.
So if it's so great, why do we need a synthetic version?
Because the natural version is incredibly unstable. It breaks down in the body in about 6 minutes.
Useless as a drug.
Totally. So researchers stabilized the structure and created MXB22510.
So what can this stable version do?
Well, the initial clinical target is chronic sinusitis, which is actually a major gateway for pathogens into the brain. But the molecule itself has broad-spectrum efficacy. It works against bacteria, even antibiotic-resistant ones, fungi, and viruses like herpes and SARS-CoV-2 in animal models.
So it's basically a systemic cleanup crew.
It supports immune health, clears pathogens, and helps with that necessary removal of damaged cells. All things you need for a longer health span.
And what about safety?
It looks extremely safe. No toxicity seen up to 3,000 times the effective dose. And importantly, it doesn't mess with the healthy gut microbiome. It's a stable, long-acting way to restore a critical healing factor that we lose as we get older.
So let's bring this all together. It's been a fascinating deep dive into the systemic nature of aging. And the synthesis seems pretty clear. Aging is driven by toxic factors like SASP, but we have proven ways to intervene.
We do. We have the subtractive path, remove the inhibitory factors with something like TPE, and the body's own youthful proteins rebound. You get measurable biological age reversal.
You're just clearing the debris so the body can heal itself.
Exactly.
And then there's the additive path.
Where you deliver this targeted restorative cargo, like the E5 miRNA cocktail, a precision senomorphic that shuts down the toxic machinery by targeting regulators like PCF and HIPK2.
So what this means for you, the listener, is that these strategies go way beyond just treating symptoms or single diseases.
They target the foundational mechanisms of aging, and they offer a scientifically validated way to extend your health span across almost every tissue in your body at the same time.
We've seen that removing the old works, and we've seen that adding the targeted good stuff works, which leaves us with a really potent, provocative question for the future of all this. Given that each approach solves a different piece of the puzzle, one dilutes the inhibitors, the other silences the toxicity switch, what would happen if you combined them?
Right. What would be the most effective intervention of all? Would it be a precision therapy that first uses TPE to clear out the systemic junk?
And then follows up with a targeted restorative senomorphic cocktail, like the E5 miRNAs, to lock in that rejuvenation effect, that potent 1-2 punch.
Yeah.
That's the combination researchers are racing to test right now.